All right. Hopefully our audience in the Okanagan is able to tune in. And I'm just going to jump right into it. So a strange inborn error of metabolism. So inborn errors of metabolism, this is a class of rare genetic disorders. And essentially, usually they are involving some kind of defect in an enzyme or a pathway. And so this case specifically was 3 unrelated affected patients with healthy parents. They were born healthy, but there was an onset of kind of progressive deterioration and ataxia. And ataxia kind of has hallmarks with difficulty walking and tremors, slurred speech. And because they were healthy parents, we're really looking for something that's recessive, either homozygous or compound heterozygous, meaning 2 hits in the same gene, or de novo variants. And so because there was suspected metabolic defect, this was followed up by some very specific biochemical tests. And so deep biochemical phenotyping really pointed towards glutaminase, or GLS, as the broken gene. And so GLS, its main function within metabolism is to essentially convert glutamine into glutamate so it can be used for energy within the mitochondria. And so within these 3 patients, there was a very high level of glutamine and a very low level of glutamate. So typically these are present in controls at about 1:1, but in our patients this was, anywhere from 5 to 10-fold higher than it should have been. So exome sequencing, it's almost routine for clinical diagnosis of rare genetic disorders, and it's really bolstered how we can diagnose these cases. And so exome sequencing, I'm sure you've all heard of it, but it's going to target primarily the coding regions of the genome, these little green exons, and it's mainly been used to find protein coding variants. So through variant calling and prioritization, which probably is its own talk, but it's not the focus of this one, we can take a sample, call variants on it, and then take those variants and annotate them. And if this is something you're interested in, I encourage you to talk to me later. But at the end of this process, we get essentially a set of candidate variants that are rare and damaging. And so after running the exome sequencing pipeline, we ended up finding single hits in GLS for only 2 out of 3 of the families. So for family 1, there was a missense mutation in exon 6, and in family 3, a frame shift that caused a nonsense mutation also in exon 6. These are very rare, and when they were flipped into HEC293 cells, they completely abolished the activity of GLS. So these mutations are wiping it out. But because we've got healthy parents, this clearly cannot be the whole story. There must be something else going on, and from a protein standpoint, we can see a greatly reduced amount of GLS in these patients from a Western blot. So really what this is kind of indicating is there could be some kind of repression going on of the maternal allele. And so what we did was for family 1, we did some Sanger sequencing of the cDNA and identified allele-specific expression that showed the parental allele was much more highly expressed than the maternal allele. So the hypothesis was there's got to be something in this region that's shutting down that maternal allele, and how do we go about finding that? Well, it's probably going to be a non-coding mutation because the exome didn't find anything. So we ended up doing whole genome sequencing, and we really kind of focused our narrow search on variants that might be in the promoter region, in these untranslated regions or UTRs, or in the introns. And to make a long story short, a lot of time in IGV, I ended up finding some really odd signal in the 5' UTR. So this is kind of upstream of where the gene starts to become protein coding. And so this odd pileup also had some signals where on one allele there seemed to be a deletion of about 24 nucleotides, and on the other allele the reads were mapping kind of in an awkward manner, such that many of the reads were only comprised of this GCA, hence my little slide image. And so that's when I noticed that this was over a short tandem repeat, or STR track, and that's when things started to get a little more exciting. So short tandem repeats, what are they? They're everywhere in the genome. They're in the coding regions, the UTRs, introns, there's intergenic STRs, and they can be anywhere from 2 nucleotides to more than 10. And this just kind of shows you the magnitude. There are a lot of them across the genome. But how do they result in disease? I can only give you a quick example. FMR1, which is responsible for Fragile X syndrome, has essentially a repeat expansion in the 5' UTR, and that causes hypomethylation, because that, or hypermethylation, because that repeat is CGG. So there are about 30 known pathogenic STRs in the genome. Most of them are related to ataxia. There's also Huntington's, which is a coding STR. But kind of finding a new STR, that's pretty exciting. So turns out from PCR-free whole genome sequencing data, there's a new tool called Expansion Hunter. These tools always have cute names, which is designed for genotyping locus-specific STRs. And so the way that it works kind of at a high level is that if the repeat is shorter than the read, then it can be exactly genotyped. If the repeat is present in flanking reads, where part of the read maps outside of the repeat and the other part of the read maps to the inside of the repeat, then you can get a pretty good confidence interval for the size of that repeat. And then lastly, if there are reads that are anchoring on the either side of that repeat or entirely composed of the repeat, then really what Expansion Hunter is going to give you is a confident lower bound on how big that STR is, which is important. The other important fact is that it runs in about 5 seconds per locus, and all it needs is a BAM file and a JSON. And so ran Expansion Hunter on this patient 1 whole genome, ended up finding an expansion of 94 GCAs. So in the genome it's 16. In our expansion, it's 94, and it is 8 on the paternal allele. And so we've now got a bit of an explanation for why that maternal allele might be shutting down. And so we used repeat prime PCR and confirmed this repeat in family 1, and it also ended up being homozygous in family 2, and it was also found in family 3. So we seem to have found the missing heritability, and because repeat prime PCR and Expansion Hunter are a bit limited in being able to detect the full length of this, we did PCR with some very tough conditions and ended up finding that this repeat is massive. The smallest one is 680 repeat copies in the patient in family 1, and in family 2 heterozygous or homozygous, and family 3 heterozygous. We also established a few really rapid collaborations based on the fact that this can run in only 5 seconds. It allows us to genotype a massive amount of genomes very quickly. And so by doing this in a population of over 8,000 individuals with PCR-free whole genome sequencing data, we ended up finding only one example of an expanded allele. So we can roughly put the frequency of this expansion to be 6 times 10 to the negative 5th. And this work so far is a part of a large collaboration. If you're interested in the mechanism of this expansion, find me and we can talk later, or I do have a slide at the back, but I've got to hit my time limit so Melissa can have some stuff to talk about. But it's bioinformatics, so I have to have a little bit of a spin on it. And if we all had whole genome sequencing on the patients from the beginning, could we have solved this quicker and without the deep phenotyping? Or put another way, how can we extend this anecdote of manual IGV inspection to scale for whole genome sequencing data? I mean, we've got 1 million or more short tandem repeats within genes across the genome. So how do we know, how can we detect these without looking at IGV? And because Expansion Hunter is a locus-specific tool, it's really not designed for that genome-wide analysis. So in comes another cutely named tool, Gangster, and it has been developed for these genome-wide capabilities. So if we use this case as our gold standard, how does Gangster perform? So Gangster, this is Expansion Hunter. We've got 94 copies and 8 copies. Gangster also has a local mode where you can just give it a single locus and it can estimate the genotype. That's calling at about 60 copies. But unfortunately, when you run gangster in its genome-wide mode, it completely fails to detect this expansion. And so this is a problem because if you had done this without knowing about this locus and you saw 8 and 21 and in your population, that puts you kind of right in the normal range. So gangster doesn't quite fit the bill. And so some next steps for this is to benchmark a few different methods. Got a collaboration with Expansion Hunter developers at Illumina. There's also a newly published tool called Stretch, and if anybody has others that they're interested in seeing benchmarked, let me know. We can benchmark this against our gold standard case, but we can also generate synthetic expansions and even synthetic novel expansions within known disease genes. Then we'll run this on undiagnosed genomes at the BC Children's Hospital, and hopefully we'll find new novel SDR expansions underlying genetic disease. And if you're coming to ASHG in October, find my poster and I will talk more about it. With that, I'd like to thank my collaborators, the Wasserman Lab, and my funding through BC Children's Hospital. And I will take questions. Yeah.
When you guys first started suspecting that glutamine pathway as whatever the driving force behind this disease, why did you decide to do whole genome sequencing instead of just focusing on that one locus for your sequencing?
So the true kind of like the way the sequencing went was there was a clinical panel ordered first that came back negative. Then we did exome, and even through exome, the standard full penetrance models that we typically run came back negative because there were only those singleton hits in GLS. And then we went to whole genome sequencing. But in a world where whole genome is done first, then, and we're perfectly capable of detecting these things, we can start to build, a full picture from the beginning, and that'll allow us to, not go back and get more DNA, more sequencing, more DNA, more sequencing, because it really just extends the time to diagnosis.
Look at the quality metrics of your raw files to basically look for a repetitive region that might sunset your search space.
So the question is, do I look at the quality metrics of the region to see if there are...
I'll give you alignment to see if any kind of repetitive region that has difficulty aligning that. Would that be used to subset your search space?
Subset the search space for possible STRs?
Yeah.
Yeah, and so that's kind of like the approach that that Gangster takes, is it looks for these, you can find short tandem repeats just based off tandem repeat finder on the sequence itself, and then you can kind of look at those loci and any types of repeats that might be present in the same kind of trinucleotide or dinucleotide or pentanucleotide track and kind of rebuild the picture. The fact that Gangster was kind of unable to genotype it at its full length while doing it in the genome kind of agnostic scale speaks to maybe, there's some new methods that need to be tried. So I think, it is definitely something that's incorporated into it is, you've got these regions that are inherently hard to map around just because they're short, repetitive sequences. But oftentimes kind of local realignment of the region can help resolve those types of issues.
Does Gangster make use of something like the chromogen and annotation of functional regions, or is it just looking at everything agnostic?
Are you talking about how you prioritize?
Yeah, does it prioritize looking at functional alignment and functional regions?
No, it's completely agnostic, but then you can always add any kind of annotation afterwards. And that's the last question I'll take. So.